In today's video, we will study the molecular structures of DNA, RNA, and proteins, and how genetic information flows through these biomolecules. By the end of the video, you shall be able to attain the following learning targets. All living things are made up of biomolecules, which are large molecules necessary for life. Examples of these includes lipids, nucleic acids, carbohydrates, and proteins. But for this lesson, we will just focus our discussion on the structure and function of nucleic acids as well as proteins. The building blocks that make up these biomolecules are called monomers. Repeated units of these monomers create long chains of molecules called polymers. Think of these building blocks like Legos. Each blocks of Legos represent a monomer to form longer chains called polymers. In nucleic acids, the monomer unit is called a nucleotide, and in proteins, it is called amino acid. Let us first study the structure of nucleic acids in detail. There are two types of nucleic acids, the DNA or deoxyribonucleic acid and the RNA or the ribonucleic acid. We can summarize a few differences between them through this table. For the first one, DNA is a double-stranded molecule, while RNA is single-stranded. Number two, DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. The difference between the two is the presence of a hydroxyl group in the structure of the ribose, which is actually absent in that of the deoxyribose. DNA and RNA complementary base pairing is slightly different with DNA and RNA. With DNA, it uses nitrogenous bases such as adenine, thymine, cytosine, and guanine. Meanwhile, for RNA, it makes use of the same nitrogenous bases. However, uracil replaces thymine. And lastly, DNA is responsible for storing and transferring genetic information, while RNA directly codes for amino acids and acts as a messenger between DNA and ribosomes to make proteins. As mentioned, DNA and RNA are made up of monomers known as nucleotides, and each nucleotide is made up of three components, namely a nitrogenous base, a pentose or a 5-carbon sugar, and a phosphate group. Now let's consider them, each of the following components, into the next slide, beginning with that of the nitrogenous bases. These bases are so named because they contain carbon and nitrogen. DNA nucleotides contain the nitrogenous bases cytosine, thymine, adenine, and guanine, while RNA nucleotides contain the same nitrogenous bases except for thymine. Instead, it is uracil that replaces thymine in RNA. Adenine and guanine are classified as purines, which has two rings in their structure, while cytosine, thymine, and uracil are classified as pyrimidines, which have a single ring as their primary structure. These nitrogenous bases are simply known by their symbols A, T, G, C, and U. Next, we examine the 5-carbon sugar. The carbon atoms of the sugar molecule are numbered as 1, 2, 3, 4, and 5. The single apostrophe alongside these numbers is read as prime. In RNA, the sugar is ribose with a hydroxyl group on its second carbon, while in DNA, the sugar is deoxyribose with only a hydrogen on carbon number 2, hence the term deoxy, meaning no oxygen is attached to it. Finally, we move to the phosphate group. As you can see, the phosphate groups presented here are in yellow circle, and it links the sugars of two adjacent nucleotides. This linkage results in a repeating pattern of sugar phosphate units called the sugar phosphate backbone. Note that the nitrogenous bases are not part of the backbone. So it is important to take note that the nucleotides are bonded by phosphodiester bonds, which links the phosphate groups of one nucleotide and the sugar of another molecule. Okay? Notice that one end of the phosphate group is attached to the number 5 carbon of one sugar, and the other is attached to the number 3 carbon of the next pentose sugar in a nucleotide. We refer to this as the 5' end and the 3' end respectively. Therefore, we can say that a polynucleotide, meaning repeating patterns of nucleotides, has a built-in directionality along its sugar phosphate backbone. On the other hand, the nitrogenous bases are attached along the sugar phosphate backbone. DNA molecules have two polynucleotides or strands that wind around an imaginary axis forming a double helix. The two sugar phosphate backbones run in opposite 5' to 3' directions from each other. This arrangement is referred to as antiparallel, somewhat like a divided highway. Let us now study nitrogenous base pairings. In DNA, adenine in one strand always pairs with thymine in the other, and guanine always pairs with cytosine and vice versa. If a stretch of one strand of the DNA has the base sequence of AGG-TCCG, then the base pairing rules tells us that the same stretch of the other strand must have the sequence TCC-AGGC. Remember, each strand is antiparallel with each other, hence the directionality runs opposite each other. In RNA, on the other hand, they do have similar base pairings. However, for thymine, it is not present in RNA. So, uracil replaces it. If a single-stranded RNA were to copy the information from a DNA template, cytosine pairs with guanine and vice versa, while adenine pairs with thymine, but uracil pairs instead with adenine. The base pairings are matched this way because adenine and guanine are purines, meaning they have two rings in their structure, while cytosine and thymine are pyrimidines which have a single ring. Pairing a purine with a pyrimidine is the only combination that results in a uniform diameter for the double helix. Now that we are done with the structure of nucleic acids, let us discuss proteins. Proteins are the functional products of genes and execute various cellular functions. Proteins can be enzymes that catalyze reactions, protect against diseases, act as storage of amino acids, or as transport proteins. Further, it can also regulate an organism's activities, respond to stimuli, aid in movement, and provide support. The monomer units of proteins are called amino acids, while its polymer is called polypeptide. An amino acid is a molecule with both an amino group and a carboxyl group. At the center of the amino acid is a carbon atom called the alpha carbon. Its four different partners are an amino group, a carboxyl group, a hydrogen atom, and a variable group symbolized by letter R. This R group is also called the side chain, which differentiates each amino acid in a protein. There are 20 amino acids that cells use to build thousands of proteins. The amino acids are grouped according to the properties of their side chains. One group consists of amino acids with nonpolar side chains, which are hydrophobic. Another group consists of amino acids with polar side chains, which are hydrophilic. Acidic amino acids have side chains that are generally negative in charge, while basic amino acids have amino groups in their side chains that are generally positive in charge. The 20 different types of amino acids can be combined to make a protein. The sequence of amino acids determines each protein's unique overall shape and its specific function. Now, there are four structural properties of proteins, namely primary, secondary, tertiary, and quaternary. The structure of protein is analogous to a long strand of a yarn. At the primary level, the yarn still has no discernible shape. As the yarns are woven together in a specific way, it starts to gain shape and function. When the weaving is done, it creates the final product, which is the sweater, hence, comparatively, it's like the protein. The primary structure of a protein is its sequence of amino acids. As an example, let's consider transthyretin. It is a blood protein that transports vitamin A and one of the thyroid hormones throughout the body. Transthyretin is composed of 127 amino acids. Each of the 127 positions along the chain is occupied by one of the 20 amino acids, indicated here by its three-letter abbreviation. Next, the secondary structure of proteins occur when the polypeptide chains form a helix or a pleated sheet structure. The alpha helix is shaped like a coil, and the beta pleated sheets have two or more segments of the polypeptide chain lying side by side together. The coils and folds are the result of hydrogen bonds between the repeating units of the polypeptide backbone. Further, while secondary structure involves interactions between the backbone constituents, tertiary structure is the overall shape of a polypeptide resulting from interactions between the side chains of the various amino acids. And lastly, for the quaternary structure, it is the overall protein structure that results from the aggregation of these polypeptide units. So, it is a multiple combination of polypeptides making up this structure. An example of a protein in a quaternary structure is the hemoglobin. Hemoglobin consists of four polypeptide subunits, two of which are alpha and another two beta subunits. Each subunit has a non-polypeptide component called heme with an iron atom that binds oxygen. Sometimes, a slight change in the primary structure can affect a protein's shape and ability to function. For instance, the sickle cell disease. It is an inherited blood disorder which is caused by the substitution of one amino acid valine for the normal one, which is glutamic acid, at the position of the sixth amino acid in the primary structure of hemoglobin. A person with this disease could have clogged blood vessels impeding blood flow. And since the blood vessels are shaped like a sickle, it does not become good carriers of oxygen necessary for circulation. We are now done identifying the components that make up nucleic acids and proteins. We will now relate how DNA, RNA, and proteins work together. The central dogma of molecular biology states that genetic information flows from the DNA to RNA to proteins. The DNA is the genetic material that organisms inherit from their parents. DNA contains the instructions needed for an organism to develop, survive, and reproduce. To carry out these functions, DNA sequences must be converted into messages that can be used to synthesize proteins. These messages coded in the DNA are transcribed by the RNA molecule. In this case, a specific type of RNA called mRNA, short for messenger RNA, carries the recipe to form precise orders of amino acids, which are the building blocks of proteins. It is in the cellular structures called ribosomes where sequences of amino acids are translated to form a polypeptide chain, which will later be formed as proteins. Proteins are complex molecules that do most of the work in our body. So, in summary, this entire process is called gene expression. The information encoded in a gene is used to direct the assembly of a protein molecule needed by the body. Let us now summarize what we've learned beginning with nucleic acids. The building blocks of DNA and RNA are nucleotides. These nucleotides are composed of phosphate group, a pentose sugar, and a nitrogenous bases. There are differences between the two nucleic acids as summarized and as listed in the table here. Meanwhile, the building block of protein is an amino acid. It is composed of four different groups, which are the amino group, the carboxyl group, the hydrogen atom, and a variable group called side chain, which may vary either as polar, nonpolar, or with negative or positive charge. Proteins have four structural levels, namely primary, secondary, tertiary, and quaternary. Proteins are functional products of genes and they execute vast cellular functions, and it is directed and controlled primarily by the DNA. That would be all for today's lesson. Thank you so much for listening.